So first things first, thank you so much to everyone who is joining us, and thank you to ECN for sponsoring this seminar series. This is our very first presentation. So I will pop some instructions in the chat in just a few seconds, but please do use the chat if you want to ask any questions. We will have a recording of the chat, and then we can ask those questions to Miles once the presentation is complete. So we can by introducing our speaker. Dr. Miles Zhang's research interests include taxonomy, systematics, and the phylogenomics of Hymenoptera, in particular, gall wasps and chalcidoids. He holds a bachelor's degree in zoology from the University of Guelph, a master's degree in biology from Laurentian University, and a PhD in biology from the University of Central Florida. After completing a postdoctoral fellowship at the University of Florida working on ants, he became an ORISE postdoctoral research scholar with the USDA Systematic Entomology Lab, working at the National Museum of Natural History. He is also a leader in the use of social media for entomological outreach and is one of the founders of the Entomology Memes Facebook group. Today, he will tell us about North American oak gall wasps and their natural enemies. So take it away, Miles.
Great. Thank you, Sandra. Thank you all for coming and listening to me to, blab on about one of my favorite things in the world, the gall wasps. I think this is a really fantastic idea to have ECN host these type of museum-based research talks, and I look forward to seeing more of them in the future. So the title of my presentation is Fantastic Tiny Beasts and How to Identify Them, Advances in the Study of Gall Wasp Communities in the Genomic Age. So here's a picture of one of the gall wasps I photographed in Florida, just emerging out of its own gall. So that's what it looks like. But often when I tell people I work with galls, they have no idea what I'm talking about, or they're thinking like gall bladders or that kind of doctor. Not quite. So galls, there's a broad definition of galls, but generally when you're talking about plant galls, they're basically some sort of abnormal growth that's caused by another organism, right? In this wonderful review by Stone Shoronge in 2003, they've categorized all these different types of galls, and you can see the definition varies quite a bit. So you have things that's basically rolled up leaves that can be called galls, some sort of invagination that's not fully closed, to like one of these gall wasp galls, which are completely enclosed. So all of these can be considered as galls. And the reason for this diversity is because 6 different orders of insects can make plant galls. Right, so these are Diptera, Hymenoptera, Hemiptera, Coleoptera, Thysanoptera, and Lepidoptera. So there's a lot of insects that have converged on this type of feeding behavior. And the reason for that is because it provides food and protection for their young. Right, so rather than feeding, like a caterpillar just chewing on a leaf, it's completely exposed to predators and parasitoids. But if you're even, just hidden from sight, even in these rolled up leaves, you get more protection that way. So, and I'll talk more about some of the more advanced galls where they even create, new sources of nutrients for them to feed on. So gall wasps are in the family Cynipidae within Hymenoptera. They are the second largest group of gall-inducing insects, the largest being the Cycidomyids, the gall midges. So there are about 1,500 species worldwide, and that's what a gall wasp looks like. And here are some galls in my hands that I collected this summer for comparison. They're either gall inducers, meaning that they create their own galls, or inquilines. So in inquilines, they lost the ability to make their own galls, but instead they basically just lay eggs into other existing galls and kind of modify them. So not completely lost the ability to change, plant tissues, but they cannot make their own galls. And the majority of this diversity in gall wasps are found on oaks and roses, but of course there's a few other ones like various herbs, so like asters and Lamiaceae, they'll have galls as well. So the reason why galls, and in particular cynipid galls, are really interesting and is the focus of a lot of these recent studies is because it can be considered as an extended phenotype of the gall wasp, right? So think of it as the extension of its gene. Because they can create these really structurally complex galls. In fact, they are the most structurally complex out of all the gall inducers. You can see some of the pictures here. They have various external and internal modifications. I'll show you more in a second. But despite all that, this process is still poorly known. We don't know how they're able to manipulate plant tissues. So now there's some recent transcriptomic studies that have looked at galls at various stages of development. There's all these genes that's being upregulated and downregulated. So there's like huge changes at different parts of the plant tissue. In fact, the gall wasps are capable of making a nutritive cell layer just outside of their larval chamber. These are starch-rich, so nutrient-rich cells that's the only source of food that they need. And this source of food is created by the gall wasps because this type of cells are not found anywhere else in the plant. So there's a lot of active research and interest in this, but unfortunately, we still know very little about this. So the external and internal modifications ranges from spines to hairs. Some of them are able to create resin or nectar that you attract ants and wasps that'll act as a protector. There's even these internal airspace. So like this one shown on the right, where they have suspended chambers that actually some of them will dissolve away, and you'll have this rolling chamber in the middle where the larva lives. And all of this is thought to have evolved as protection from natural enemies. So here are just some diversity of the galls that I've photographed over the years. The one on the bottom right is not by me, but I love this photo. So this is an oak gall wasp. If you live in North America, you probably have seen these. You open them up, they have that suspended chamber in the middle. It's completely suspended on all sides by these fibers. So it's just really neat, and they're quite big, right? Like the ones I had in my hand, they can reach quite a large size, but the wasps are relatively small still, less than 5 millimeters, and that's about as big as gall wasps get. Okay, so like I said, there's a lot of natural enemies associated with galls, and when we study them as a whole, we call it the gall wasp community. All right, so here's a host plant, and then you have an inducer of the gall. And then you have these inquilines, right? So these are also cynipids that are capable of modifying the galls. So you have inducers, inquilines, and then you have various parasitoids that will attack either of these two. And then you'll have hyperparasitoids that attack the parasitoids. So you have basically a 3-4 level, you can go up in the trophic level. And all of this resource is created by the gall wasp themselves. So you can consider them as an ecosystem engineer because they are creating and sustaining all this biodiversity. And this has been used as model systems for a lot of evolutionary studies, population genetics, genomics. You're trying to figure out, the demographic histories. There's a lot of cool studies associated with these systems, but the majority of this are all done in Europe, where the system is well known. And I'll get back to this in a little bit. But yeah, so here are just some more recent references. If you're interested in these types of research, just search for galls and like ecology or evolutionary biology, and you get all kinds of cool research associated with it. But in North America, the fauna is poorly known, but it doesn't mean that nobody has worked on it. So one of the most influential figure, one of the first famous taxonomists of gall wasp is Alfred Kinsey, and you might know him as the sexologist, which he worked on human sexuality and later in his career, but the first 20 years of his life, he's actually dedicated to gall wasp research. In fact, that he's done so much collecting in the US as well as many parts of Mexico. His specimens, I think, ranges up to 9 million individuals, are all stored at the American Museum of Natural History. And in fact, that he actually came up with one of the first phylogenies of gall wasps. And this was, this is 1920. So this is way before the times of Willie Hennig. who created the whole cladistics thing. So it's really cool to see this type of rudimentary trees where he's thinking about the relationships between these different genera of gall wasps. If you're interested in his life, is there actually a movie about him played by Liam Neeson? So, and in fact, there is a little bit on the gall wasp. So I thought that was pretty interesting. So do check that out if you're a fan of Kinsey or Liam Neeson. Another historical figure is Lewis Weld, and he's wealthy, independently wealthy, so he never really had to hold down a job. So he just spent basically most of his life just traveling and collecting galls and describing them. So it sounds pretty cool. And the majority of the types are here at the Smithsonian. So I actually have a few right behind me that I'm looking at right now. So it's a fantastic resource that he's done, and this was in like in the 50s, 60s, up to 70s, and he's created these 3-volume texts that we still use to this day, basically dividing the gall wasp of the U.S. into the Pacific slopes, the Southwest, and Eastern North America. Here are just some of the figures that he had, which I guess were pretty good for its time, but of course it's kind of frustrating when you get to certain pages. The photos are not very clear, but unfortunately this was still, for many of us, is still the best resource that we often refer back to constantly. But after Lewis, there's not much work done in North America. There are a few here and there, but nothing like big scale taxonomic revisions. In fact, most of the modern gall wasp research are done by European researchers, the active field research. So those studies that I've shown you earlier, those are all done mostly by Europeans. And so here are just a few of the people. You might recognize some of them, Frederick Rundquist, for example, who's also created Mr. Bayes, if you're into phylogenetics. So he's also a Bayesian phylogeneticist, but also really interested in gall wasps. But by comparison, many of these other regions are incredibly understudied, and that includes North America. So this is one of the first studies, molecular studies published by Rundquist et al., including many of the authors that I mentioned. And this is a multilocus phylogeny. This includes 5 genes plus morphology. And then they're using ebaliids, which are these like big cynipoids that's related to cynipids, using that as an outgroup. And this is the tree that they got. It's a little confusing with all these arrows and colors, but basically what they're showing is Cynipidae is monophyletic, right here shown in green. That means that they have one common ancestor, and then the inquilines have showed up twice, but they're distantly related. Sorry, actually 3 times, but distantly related. So what this means is that gall, if that's the case, if Cynipidae is monophyletic, that means that gall induction have only evolved once. Their ancestors evolved this, and then all the inquilines have secondarily lost this ability, right? And for a while, and this is what we agreed on, and it seemed to make sense, taxonomically, they've always thought to have been monophyletic. And that is until just 2 years ago, Bonnie Blamer at Smithsonian now in the Berlin Museum, along with Matt Buffington, who's the curator of cynipoids here at the Smithsonian, they've generated a UCE phylogeny. UCEs are ultra-conserved elements, which are basically really conserved regions of the genome that you can get across, like you can get these genes across the genome. And they did this for the cynipoids as a whole, and they got over 1,000 loci on average from museum samples, right? So that's another advance in these type of molecular techniques. So rather than just using relatively freshly collected samples that with the perfect DNA or like hardly degraded, you're able to go into museum collections and then start getting DNA from much older samples. And because they're looking at cynipoids as a whole, they actually got another superfamily, platygastroids, as the outgroup, right? So that's different than the previous study where they're using another cynipid as the outgroup. Here, they're using a different superfamily. So, and here is the resulting tree. But what is immediately different about this one, don't worry too much about the colors, is that Cynipidae is no longer monophyletic. In fact, it's polyphyletic. So on the bottom where it says Cynipidae, those are, I guess, the core cynipids. But then you have a few groups that have actually popped out in different parts of the tree. Some are more closely related to other families. Some are actually, basically basal to all of cynipoids. So that means that they're more distantly related than all of the other cynipids. And this actually includes the rose gall wasp, which I spent a lot of time studying. So this is really interesting, which means that gall induction have evolved multiple times. And eventually, if this holds true, then some of these families that's currently being grouped under Cynipidae should be named as distinct families of their own. Okay. But today I'll not be talking about any of the other groups. As much as I love the rose gall wasp, today I'll be focusing just on the oak galls, the Cynipini. So they are the largest group, and they induce galls on Fagaceae, right? So this includes oaks that probably most people are familiar with, but also related genera such as chestnut, chinquapin, and a few other groups. They're also the largest tribe, and in fact, they're about 1,000 described species, so that's about 2/3 of all known cynipids. And they're currently grouped in 51 genera, but taxonomically, you can only describe them as chaotic because almost every genus is para to polyphyletic. It's really, once you get to Cynipini, getting it down further to even to the genus level, it can be a headache. And just to show you a little bit of the amount of change that has done. So Melika and Abrahamson in 2002 revised this Cynipini, and then they recognized 26 genera. But since then, it's only been 20 years. 8 of those have been re-established. So people disagreed with their synonymization, and then they re-established it. And then 17 new genera have been described. So in fact, I just reviewed a paper that they're re-establishing another one of Kinsey's old names, so it's probably more coming down the pipelines. But so it is rather chaotic, but I guess it's in a way it's exciting because there's a lot of we're actively advancing the knowledge of this group, and especially for North America, because North America we have about 700 known species, so that's 2/3. Okay. But there's a reason why they're difficult to work with, and that starts with their host plants. The oaks themselves, they're pretty messy. Oaks are divided into 2 subgenera, 8 sections, and over 500 species worldwide. And in North America, we have about 200 of those. And this term, I actually never seen it before until I started reading oak literature. They call it a syngamion because basically there's so much hybridization going on with members of the oaks, at least within, sections within the oaks, that it's like unlike anything else. So here's a phylogeny. So if you're familiar with phylogenetic tree, this is not a tree, this is a network, right? So network kind of shows the hybridization events going on. So you can just see how messy it can get. And identifying oaks in the field can really get tricky because there's all these hybridization with intermediate characters. You don't know which one is which. But seems like gall wasps are pretty good at identifying them, at least down to specific sections. Most gall wasps are section specific, so it means that if they are specialists on red oaks, they will not they will not be found on any of the other oaks. And this pretty much holds true for everything we know in North America. In Europe, there are some that switches between 2 different sections, but those are relatively rare, and we don't have any of that, to my knowledge, in North America. And cynipids also practice, the oak galls have this thing called cyclical parthenogenesis. All right, so where they alternate between sexual and asexual generations. So what does that mean? So here's a diagram from Tatsuya Ide and that's Tatsuya Ide is actually one person. I just misspelled, like I put his first and last name together. So this is Ide and Abe's research. So here's showing in blue is the sexual generation. This occurs in the spring, where there's males and females. They will come out of these leaf galls, and then the female, they will mate. The female will lay eggs into the leaf. They will develop into these leaf galls that looks different, right? And then in the fall, this is the asexual generation. So only females will come out, and these females look different than the sexual generation males. Oh, sorry, the sexual generation females. And even the galls look different, right? And then they oviposit into the leaf buds, and then the cycle repeats itself. So as you can imagine, if the adult and the gall morphology looks different between generations, taxonomy-wise, it's a bit of a nightmare, because you have no idea which one links with which unless you have detailed rearing records. At least back in the days. Now we have DNA, which helps a little bit. But so for a lot of the 700 species that we have in North America, we only know them from one generation. So it could be that we just haven't discovered the other generations. And this is often the case with the spring generation, which tends to be on like really ephemeral resources, such as the flowers, like the catkins of the oaks. They don't last very long. They just come out, make galls, die. So we don't know a lot about those sections. But worse is that in some cases, they're described under different names or even different genera. And we'll try to link those together has been, a major problem. So like I said, there's of course DNA, right? So, but it doesn't work all the time. And so here are just in one example that I recently revised this genus, Belonocnema. And here's on top is the spring generation. So this is the sexual generation. That the one on the top right is a sexual female that comes out of these potato-like galls that's on the root of live oaks. Whereas on the bottom is a leaf gall. This is the asexual generation. And here is this weird little wingless, not wingless, but they have these twisted wings that they cannot fly. Instead, they have these large tarsal claws on their front leg that they use to dig into the soil. And then because they come out of these leaf galls, they go straight under and they start making these root galls. So you see that they do look quite different. And this is one group that I started revising because we realized there's some morphological differences. I'm not going to get into too much about the taxonomy, but basically, since its description 160 years ago, there's a lot of confusion on whether there's one species or up to 3 species. So we did CO1 barcoding. This is mostly done by my collaborators in Texas, Scott Egan and Jim Ott. Right, CO1, the DNA barcoding is pretty standard in taxonomy, since like it's a universal gene, or so we thought, that you can use to distinguish species. But here I have the three species that morphologically distinct, colored here in orange, green, and purple. And you see on the tree that the purple and the green are not distinguished, even though morphologically they're quite distinctive. And then we looked at the same data set using genome-wide single nucleotide polymorphisms, which are nuclear gene differences, right? So, and then here you see that all 3 of these colors separated out nicely. So, and this supports the morphology. So there are indeed 3 species, but CO1 was not able to recognize 2 of them. So in these cases, they're called mitonuclear discordance. So there's just, for whatever reason, it could be at the rate in which these genes are evolving at different rates. So CO1 is just not very good at distinguishing these species. And this has been known recorded in Europe as well in some of the early days. So in fact, they don't do CO1 at all for cynipids. At least the oak cynipids. They use Cyt B as a mitochondrial gene, and then they use a few other nuclear genes. So for the people that's strictly relying on CO1 some people are even suggesting using that as describing new species just based on barcodes. Well, here's an example why that will not work. So So there are definitely some challenges in working with Cynipini, the oak gallers. In fact, one of the major problems, of course, is the higher phylogeny. We need to redefine the genera because right now it's just unworkable. Everything falls into like these few trash can genera like Androcus or Caloritis, and they're essentially meaningless. And this can be done with these type of UCE techniques that Bonnie did 2 years ago, which utilizes museum samples as well as fresh samples, right? So we can go and collect all these samples, but some of them are relatively rare or used to be abundant. Now they have much more restricted range. So utilizing museum samples, it's fantastic for that because you can go maybe even sequencing. I haven't tried it myself. You can even try sequencing some of Kinsey's types. I just saw a talk by Nicholas Wahlberg yesterday at Lund University, where they're trying to, do whole genome sequencing of old, not holotypes, but paratypes of these Drosophila from the 1800s. They were able to get, the DNA are pretty degraded, but they're still able to get some information. So it's really exciting field of like museomics, right? So using museum samples in exciting ways and not non-destructively too, which is always good, right? Because there are only so many Kinsey specimens. We just keep grinding them up, or we're going to lose them all eventually. All right, so the taxonomy definitely needs work, and then just to also to close the life cycles, right? So that's basically finding the alternate generation of the known species. But in order to do all of this, we definitely need accurate rearing records, right? So going through the literature, because the oak taxonomy is so messy, the names changes a lot, combined with the mess that is oak gall wasp taxonomy, it's almost like trying to solve a murder mystery, trying to piece all these clues together. If you have museum samples, or if you're interested in collecting gall wasps, definitely record the host plan. Of course, Corcus is difficult to identify, but try your best, at least get it down to the right section. Or if you can, keep some of the leaves and keep the galls themselves, because the galls are also useful in when we're describing new species. We try our best, if we have the galls, to describe the galls as well, because they're often species specific, or at least close enough that you can identify a few closely related species just based on the morphology of the galls. So here's one of the recent work that we've been working on this for quite a while now. It's led by Anna Ward at University of Iowa. She's a PhD student with Andrew Forbes. We're looking at the phylogeny of the North American oak gallers. And this is a UCE phylogeny, just like the Blamer study. So we've included a bunch of these freshly collected Nearctic species that's mostly collected in the Northeast. That's where like surrounding Iowa regions, I guess Midwest to Northeast. But we also included samples from Texas, Arizona, and California whenever we can. And the nice thing about using UCE is because we're using the same probe set, that we can actually take some of Bonnie's data and incorporate it to our own. And what we have noticed immediately is that there are multiple shifts between these different host clades, right? So like I mentioned before, the oaks are divided into multiple sections. And there are, when you look at the phylogeny, you see that, closely related species, some are specializing in red oaks, the other ones are in white. So that denotes a host shift between these clades. Right, so we're looking at this is actually more than we previously thought. So that is pretty interesting to us. So maybe they are, like at least throughout history, are more flexible than we previously thought. But this is a work in progress. So I'm hoping to share this at least in publication form in the next month or two. But another thing about this project, we built our first phylogeny, and what comes immediately after, at least to me being a taxonomist, there's so many potential taxonomic projects, because we're using UCEs that we can also pull out, various Sanger sequences. We quickly noticed that there's a lot of immediate taxonomic issues that can be solved. right? So this is why the vouchers are important. The voucher specimens, actually, I just got them today, so Andrew sent it to me, so I need to mount these and put it in the museum and also at the Frost Museum at Penn State, where they're actively doing cynipid research. So we are hoping in the near future to close the life cycle. So here are two amphibolus that are actually alternate generations. Right now, they're under two different names, so they should be combined. These are actually under two different genera. So Ferroteras is the wingless one on top, and then Dryocosmus is the one on the bottom that's winged. So these two also have identical sequences. So and one's only known from spring, one's only known from fall. It's like, well, actually, they're the same thing. Or species-wise, we elevate subspecies and synonymize others. Kinsey, as prolific as he was a notorious splitter. Everything he collected basically had like 100 varieties that he described, right? So basically they're like subspecies. So these two is one from California, one's from Texas. These are the jumping gulls, if you're familiar with those. They're like tiny little sesame seeds that hop on the road. So these are actually two distinct species. So eventually somebody, maybe me, but more like someone else, will have to, work through these. But it was really exciting to, start seeing these patterns. It's not just an impossible mess that we've been staring at for the past 100 years. So with these molecular techniques, this helps a lot, right? But that's not all, right? So UCE is useful on its own, but it's still mostly restricted to phylogenetics, right? So here in this study by Pedro Brandão-Dias, which is one of Scott Egans's PhD student at Rice University, they were rearing other galls, and they actually quickly noticed that there's one other species that looks slightly different, and then they sequenced this, the genes are also kind of different. They're really indescript. So on this picture, you have on top in the black, that's the catkins, right? So they make galls in the catkins, and then the alternate generation makes galls in leaf nodes, right? So like in the twigs, right by the buds. So they're really small in the genus Neuroterus. So we describe this as a new species, Valhalla, named after, apparently it's like a bar at Rice University where this was collected. The tree was just outside. So there's still a lot of urban biodiversity to be found when it comes to gall wasps. But this is also, as far as we know, the first case where a new species is described along with its genome. So we sequenced the entire genome along with the new description. This just came out, I think, last week in Systematic Entomology. So the genome is 1.1 megabase pairs. It's the smallest known genome for Cynipini, right, the oak gallers, but it's still much larger than some of the other inquilines, and I'll talk more about that in the next slide. But because we have a whole genome, we can play around with it. So I was able to extract UCE loci from this genome, and then we added it to Bonnie's phylogeny. And here, again, we show that this genus, Neuroterus, like many other cynipids genera, are paraphyletic, right? So here on top, you have Neuroterus quercus baccharum, which is a Palearctic species. It's way up there on the tree, whereas Neuroterus valhalla, this new species that we described, along with another Nearctic species, Neuroterus distortus, are quite distantly related to the other ones. So I think the ones in North America will have to be moved to a new genus because Neuroterus was it's originally described as a Palearctic genus. So somewhere down the line, we'll have to work on that. But like I said, the genome, you can start asking a lot more questions, right? So here's the great study by Hodling et al., where they were looking at, the advances in insect genomics, genome sequencing, right? It's a field that's rapidly evolving, and there's a lot of genomes that's being sequenced for every group. But what's immediately interesting to us is Cynipini, they have relatively large genomes within Hymenoptera. In fact, they probably have one of the largest in Hymenoptera, despite, their size are obviously not as big as some of the other ones. So, and their genomes are way larger than the inquilines. So there's something weird going on here. So it's like, why do they have large genomes? we thought it could, one possible explanation could be the switching of lifestyles from a parasitoid to Gall induction. Maybe you lose some of these information, for lack of a better term, but that's not the case. There's one Japanese Gall wasp that's an inquiline that have secondarily regained the ability to make galls. This is Synergus itoensis, and they've sequenced the genome for this species as well because they're interested in this difference. Because every other Synergus are inquilines except for this one. So there are some gene differences, but their genome size is not necessarily bigger. So this switch, but then again, for this Synergus, this Japanese Synergus, it's a switch between basically making galls to modifying galls, right? It's not a complete switch from parasitoids to Gall induction. So we still don't know much about this, but hopefully we can compare this with some of the Gall-inducing chalcidoids. So here's a new genus of a eulophyte that makes galls on rhododendron in neotropics that I'm helping to describe. And then there's a lot of Gall-inducing chalcidoids as well, and they also evolve multiple times. So it would be really cool to look at these comparative genomics between the Gall-inducing chalcidoids versus the cynipids. But our current best theory as to why cynipids, the Cynipini, have large genomes is partially because of this cyclical parthenogenesis lifestyle, right? You're essentially cramming two organisms into one. They are distinct morphologically, they make different galls. This is our best theory. We're not sure, but it will be cool to test with the maple gall wasps, which are only found in Europe. And they make galls on maples. And so in that phylogeny by Blamer et al., it's actually one of the groups that might not even be a cynipid after all. So it might be its own family. But this is also another group that have this cyclical parthenogenesis. So anyone in Europe interested in sequencing this genome, go for it. I want to know the answer. Okay, so that's all I'm going to talk about for the gall wasps. Now moving into more of the communities. Like I mentioned before, there's a lot of different trophic level interactions going on in this system. So the one that I have been working on is in the family of Eurytomidae, particularly the genus Sycophila, which includes about 40 Nearctic species. And the one and only publication that looked at the taxonomy is published 99 years ago. So you as you can imagine, it's kind of outdated. But Sycophila, at least in North America, they're mostly associated with cynipid galls. When you move into the neotropics, as well as just southern hemisphere in general, they're more often associated with figs. But in North America, they're mostly associated with cynipid galls as parasitoids, but there are some that are associated with other chalcidoid galls as well. The 40 species are largely distinguished based on these colorations and wing bands, right? So you can see in these photos, they seem pretty distinctive, right? Some are completely like yellow, orange, some are black, some have like a variation of the two, and then the wing bands ranges between a little stigma to like these huge wing bands. And that's why I decided to work on them. I thought they were pretty. After working on Eurytoma, which is a related genus that's mostly black, I was like, oh, this will be great. This is pretty. This is easy. It'll be a quick publication. How hard could it be? Well, I was really, really wrong. It turns out there's a lot of intra or interspecific variations going on. It's really hard to delimit that boundary, right? So it seems that collected from different populations, or even the same population, the color banding changes quite a bit. And depending on the size of the organism, that also changes. So some of the smaller organisms might not have the same features as the larger ones. So I've been banging my head against the table on this group for, I guess, 3 years now. And it's nowhere near completion. But we are able to look at some of the ecological differences at least, right? So here is a CO1 tree that I'm showing you, but we've also done a subset of that with UCEs, and we're using various molecular delimitation techniques to see how many putative species there are. Now, of course, most of these methods are probably oversplitting, but right now we have 35. The real number are likely quite a bit lower. But whether this is true or not, we collected them from 44 different oak types on 18 different oak species across North America. By that, I actually mean mostly US. Sorry for the bias. I moved to the US for 6 years now. Now I'm talking just like the Americans. The world doesn't revolve around America. Yeah, so we were interested to see whether they're generalists or specialists, right? So each of these putative species, how many different oak galls do they attack? Well, it turns out that most of them seem to be extreme specialists. And we define that as they only attack a single host, whereas, and then there are 24 of those are extreme specialists. And 10 of these are specialists, have hosts ranging from 2 to 11. And then we only have one that we considered as a generalist because it had 11 plus hosts, different hosts. Of course, this is could be partially due to our sampling bias, but this is the data that we have to work with. So until we know more, this seems to hold. And what is interesting is that the parasitoids, these Sycophila, are capable of attacking galls across oak sections, right? So like I mentioned before, the oak galls, at least within different genera, they don't move around all that much. So if they're if like members of the Amphibolyps are only found on the red oaks, they're never found on the white oaks, but the Sycophila could be attacking galls on red oak, white oak, live oak, whatever. So it's more like opportunistic, so that it's just whenever they come out, whatever's around, they will attack. But so here's a picture of one of these Sycophilas ovipositing into a gall. But we're also interested in, because like these galls have all these evolutionary modifications that's thought to have evolved to deter parasitoids, right? So we wanted to see if there's specific traits that the parasitoids are honing in to overcome these evolutionary defenses that the galls have. And this is where we have to go into the literature and go at the collection, because then you're looking at the galls themselves, right? So of course you can check the literature, that's quite variable. Some, more modern descriptions have much more detailed gall descriptions, but then the older ones are like 1 or 2 lines. It's like, it's round, it's brown. It's like, wow, thanks. That doesn't help, right? So, but luckily, when you have the collections with galls, such as what we have here at the Smithsonian, you can measure and score these traits, right, based on collections. So again, if you do collect galls, please keep the galls themselves. Those are useful. But then we quickly realized there's a lot of terms being thrown around these days about galls, right? Like often describing the same thing, and standardized terms is needed, right? Like any taxonomist will know that, like when you work with a different group, just in a different family, or even depending on the author, there's various terms. But luckily, Andy Dean's group have been updating this glossary of gall terms, and it's been really helpful in categorizing some of these traits. Right, so now that we have this trait, what we did is do a PCOA, so like a correlation matrix. We basically plotted all these different gall traits on a 2D plane, right? So the stuff that's shown in red are basically all these different traits like splayed out on a trait space. And then the blue triangles are each of these different gall, these parasitoids. And which one, depending on which ones they attack, they will fall into a specific quadrant of this trait space. So then we can see if there's any patterns here. Well, it seems like distantly related Cycophila can attack galls with similar traits, right? We determined this using the phylogenetic tree. If they're closely related, so they should have shorter genetic distance from each other, but then they're attacking similar galls. So what this means that they did not co-evolve with their hosts. It's not like, species parasitoid A divided into B and C as its hosts divided into B and C, right? So this is more of a convergence, and they're converging on specific traits. In this particular case, smaller gall size and less defensive traits. So you see these galls are just slightly bumpy. They don't really have, these fancy hairs, spines, resins, and all those characteristics, or even those internal ones seems to be really good at fending off predators. But of course, this is just a single parasitoid, and then there's a plethora of other things. And some of these have longer ovipositors, which thought to, you'd be able to reach deeper into the gall. So as the galls grow bigger and harder, Cyclical might not be able to get to it, but this Ptorimus can certainly still get at that. So we want to look at this across all these different gall wasps and just to see if there's any ecological patterns. So this is a work in progress with a whole group of people, mostly from Andrew Forbes's lab. So his lab have looked at Synergis or Myrus. Now I'm looking at Sycophila, but we're hoping to do that for basically all of these different communities. So just to summarize, we've come a long way since the days of Kinsey. That was published in 1920. Bonnie's paper was published in 2020, so 100 years later. The technology certainly has advanced a lot, and we know a lot more about gall wasps, but there's still plenty of unanswered questions. And fundamental taxonomic research is still much needed in this group because we can't do any of the fancier stuff that we want, the really cool evolutionary answers, if we don't even know how these things are related to each other or what they're called, right? And even though these technological advances expedite this process and we can get these museum samples and get DNA out of them, you cannot replace collections, right? So you got to go out and collect or curate these samples. So if you are working at a collection, definitely take a look at your gall wasp collection. If you have these galls as well, try to match them or like at least just keep them until an expert can get to them. These are all valuable data, right? I still haven't been able to visit the AMNH because of COVID, but I'm hoping to do that soon. Apparently the 9 million specimens that are like spilling out, they don't even have places to put them in proper collections. So I'm looking forward to seeing that. But ultimately, I hope I've convinced you that galls are cool, and then you can actually help us find more because there's, depending on where you live, there's these fantastic resources that can help you identify some of these plant galls. But also iNaturalist is fantastic, right? So here are the various projects on iNaturalist, such as Galls in North America. And in fact, that like many of these recent discoveries are done by naturalists or naturalist-led, right? So this is the Belanocnema, those live oak galls that I've been talking about. You can see a really accurate modern distribution of these galls. And so we only know them based on our specimens from the US, but you can see it dips down to Mexico, which is not surprising at all. And these are all, records by various people that, because unlike many microhymenopterans, the galls are really distinctive. You see them and you want to know what they are, so they put it on iNaturalist. And this is all curated thanks to volunteers as well. Right, but then there's also these rare ones that's really interesting that pops out because, like, here's the original description of this Caloritis tubicola. It's like, wow, they're all over DC, and he collected so many, right, Olstensacken, but I cannot find a single one. I live here, and in fact, if you look on iNaturalist, there's only a single record now that we know of, and this is in South Carolina. So that the distribution of this species certainly has been reduced, and there are probably some that have even gone extinct that we don't even know anything about it. So, and there's, like I mentioned before, there's a lot of urban biodiversity to be discovered. So with the help of one of the community scientists, Kimberly Sasson, we are describing a new species found in Texas on an endemic oak, Quercus lacei. And in fact, that she's actually helping to train some of the grad students, undergrads in Erica Newman's lab, UT Permian Basin. And they're also looking at a different endemic oak, and they're also finding some really interesting things. And I'm looking forward to see the specimens. So they're definitely, if you're willing to look, I guarantee you'll find something, either a new record, but probably even like new species, even in North America. Right, so, and of course, I can't end without thanking this Gallformers.org, which is a fantastic resource that's completely started by community scientists. And it's an up-to-date database on all Gallformers in the US and Canada. So not just Cynipid galls, whether they're midge galls, fly galls. And then if they have things that's undescribed, like this one, and they put it like a, placeholder name. They have all these traits associated with it, distribution, iNaturalist record. It's a fantastic resource. I love it. So I just want to end this by thanking all of my collaborators. So here are just some of the ones that have helped me with this research that I've talked about today. But of course, there are many more. And then there's upcoming research from different groups, not just in oak galls, but in herb and rose galls as well. So I'm happy to see that there's an active community, and then you see that the faces are no longer just old white men. At least to me, that is a sign of progress. So with that, I would just like to thank all of my collaborators and various funding resources. And with that, I'll be happy to take any questions. Thank you.
Great. Thank you so much. The first question is from Chris Grinter, and he asks, what's the best way to preserve leaves and galls? Over time, they seem to bounce around in trays and break down. I've seen that as well, even if they are carefully pinned or braced into place.
Right, yeah, so here at Smithsonian, I think they've tried a few things, but they even have like these little, I don't know, these plastic containers that they just put the galls in there, and then they pin those into the, of course, you know, like some are easier to pin than others, but yeah, like if they do roll around a lot, I would recommend like a different smaller container that you can put them in individually or a few at a time, and then put that in the unit tray instead. That's what I've seen. I don't know if anyone else have tried something different. Yeah, like it also depends on if you collect it with the host plant, right? Of course, some of the stems you can only brace those in, but if they're just rolling around, it's best to just keep them in some sort of closed container. Yeah. If I find any more, I'll let you know, Chris.
Thank you. And so the next question is, do any wasps alternate host with life cycle phase, as do some rust fungi? Is there an interest in or research on host resistance mechanisms, non-host resistance, cultivar resistance, and mechanisms for those?
Yeah, okay, so I'm not sure what you mean in the first part, like alternate hosts with life cycle phase. So, yeah,
so maybe
I'm not
for
sure.
the, sorry, I think that might be for like the cyclical parthenogenesis, perhaps?
Yeah, so like the oak galls, they do alternate between the asexual and sexual. So I don't know much about rust fungus. Oh wait, yeah, they do have that as well, I think. But as to the research on host resistance, I think there are talks about people looking into that. I don't know any, because like in places like Colorado, I think that there's one particular gall, Dichocaspis quercus mama, that's often in like ornamental trees within the city, and it's a bit of a like horticultural problem. And then that seems to be related to certain varieties of these trees that we plant, you know, in urban environments. But I don't know if anyone is currently researching this. I know Crystal Cook, who's at Colorado, like she mentioned it to me, and maybe this is something that she's interested in looking at. But if anyone else know more or wants to look into that, by all means, the more gall research, the better.
Great, thank you. Yeah, we have comments here that there are aphid gallers that will alternate hosts, just like wheat rust does. So Hamamelistes go back from witch hazel to birch and back and forth between those two hosts.
Oh, okay. Cool.
So, yeah.
I'm getting a few messages directly to me instead of in the chat. So like I got questions for that as well. Do you want me to answer those as well? I don't know if you can see it. Okay. So Paul asks, can you recommend any website and resources for galls in Middle and Central Europe? I think there's a plant parasite of Europe that has a bunch of the gall pictures. I don't know the website off my head, but yeah, if you just search plant parasite, I think you'll find it. There's a lot more gall resources in Europe, but of course that depends on the country. I know in the UK they even have like a Gall, or like plant Gall club or something. They're very active. I don't know about other parts of Europe, so I guess that's not really central. So sorry, I'm not sure. Okay, so Ronald asked, read somewhere Gall induction involves stem cells. Where does that hypothesis stand? Not that I'm aware of, right? So it seems like, at least based on my reading of the latest ones, that they're modifying the plant tissue. They're like breaking down cell walls at first, and then they're starting the whole process. And then there's no like bacteria or anything else like, you know, like polydnavirus in like Barcodes and Ichneumonid, which are often used to subdue their hosts. There's nothing external like that. So it's just various enzymes that's being activated. But like I said, we know very little about this. So who knows? Okay. Yeah, if you have questions for me later, you can always find me on Twitter or just send me an email. Or yeah, so actually we have a Slack channel as well for, you know, people are interested in collecting galls. That's associated with Gallformers. So if anyone is interested, just send any of us a message.
Okay, and there's one more question that just came in from Paul. I'm not sure if you can see this, but the question says, you mentioned the Japanese species that's gone from parasitoid to galler to inquiline to galler. How did they figure that out? And it also says, great talk, Miles.
Right, so they are in the genus Synergus, right, which are all inquilines, right? So, and we know that the ancestor of those inquilines are Gall inducers, right? So to be a Synergus, you must have like to switch from being a Gall inducer to an inquiline. That's like a prerequisite to be Synergus. But then this species have switched back, right? So when they build a phylogenetic tree, you see that this species fits perfectly within the rest of Synergus, but that is the only one that have regained this ability. So we know that this is like a reversal of inquilism, now back to Gall induction. So that was the same when we build these phylogenetic trees. We can do this ancestral state reconstruction, so we can figure out what the ancestor traits are. And that's why it's important to sample them across these taxa, so we can accurately figure out what the ancestral trait for many of these groups are.
Great, and so we have another question in the chat from Laura. Is there any epigenetic changes associated with the Gall formation?
I don't know. I don't know if anyone knows. Like a lot of the changes or based on the recent studies are, like I said, it's like modification of the leaves, like but epigenetics, I don't think anyone's looked into. I love how whenever I give these talks, people will be like, hey, have you looked at like microbiomes or epigenetics? Like, no, we're still at the very beginning. But if you're interested in the system and want to look at specific questions, maybe find one or two of your local galls and start digging into that. I bet you'll be the first one to answer these questions. So by all means.
All right, wonderful. Well, we are at the 1 hour mark, so I think we're at time, but thank you again, Miles, for a fantastic presentation. Thank you to ECN for sponsoring this seminar series, and thank you to everyone who attended.
Thanks. Oh, it seems like people are saying that they're only able to send messages to me and other hosts. So instead of public, so maybe that's why.
Yeah.
For next time, I guess.
Oh, okay. Yes, all right, I apologize. I'm not sure how I did that, but that's definitely my fault. And next time I will fix it.
Yeah, glad to be the guinea pig.
All right, thank you.
Bye, Sandra. Thanks, everyone.